Hello, it's my pleasure to introduce our new paper published in iMetaOmics, named Transcriptomic and Metabolomic Responses of Maize Under Conventional and Biodegradable Microplastic Stress. With the increasing production and application of plastics, microplastics are ubiquitously detected in various environments. The increasing accumulation of microplastics in agricultural soils potentially threatens crop safety and quality. To resolve the intractable microplastic pollution problem, biodegradable plastics are developed and promoted as the ideal substitutes for traditionally petroleum-based plastics. However, biodegradable plastics would disintegrate into microplastics more rapidly than conventional plastics, which may result in more substantial negative effects on soil ecosystems. Therefore, it is vital to understand the effects of conventional and biodegradable microplastics on agroecosystems for more sustainable use of plastic products. Highlights: First, microplastics triggered antioxidant defense system, especially under biodegradable microplastic exposure. Second, microplastic exposure induced plant hormone signal transduction and mitogen-activated protein kinase signaling pathways. Third, the effects of microplastics on plants resulted from changing the soil environmental variables or altering the soil microbial communities. After 60 days of exposure to microplastics, only PBS microplastics significantly decreased the fresh mass and diameter of the maize. However, all microplastics triggered the antioxidant defense system of maize. To obtain the underlying insights into the responses of maize to the microplastics, we conducted a genomic transcriptome perspective on the leaf samples. The results showed that the conventional and biodegradable microplastic treatments were separated into distinct clusters, indicating varied gene transcription after these treatments. To understand the biological significance of DEGs, we carried out the functional enrichment analysis based on KEGG annotations. Benzoxazinoid biosynthesis, phenylpropanoid biosynthesis, and photosynthesis antenna proteins were the most common response pathways identified when maize was exposed to microplastics. In parallel to the transcriptome profiling, we conducted the metabolic analysis of maize leaves using non-targeted metabolomics to identify the differences under microplastic treatments. The metabolomic profiles were explored using sparse partial least squares discriminate analysis. The score plot of SPLSDA analysis indicated that PBS microplastics and other treatments had a clear separation based on the global effects of changing metabolites. The key metabolic pathways were further identified through the KEGG enrichment pathway analysis. As shown, the phenylpropanoid biosynthesis pathway was enriched under different polymer treatments. In order to further understand the mechanism of microplastic stress on maize, the study integrated the transcriptome and metabolomics results. The results showed that phenylpropanoid biosynthesis, phenylalanine, tyrosine, and tryptophan biosynthesis, and glycolysis/gluconeogenesis pathways were the main response pathways. The content of genes and metabolites in these pathways changed, indicating that microplastics may affect the energy synthesis of maize, change the content and composition of lignin, and thus have an adverse effect on plant growth. It is anticipated that the below-ground characteristics would play important roles in driving the changes of plant performance. As shown in Figure 5, the maize biochemical features and metabolome were highly correlated with soil DOC concentration, DOM fluorescent components, the community evenness, and the relative abundance of the phylum Mycota. The biochemical, metabolomic, and transcriptomic responses of the maize to microplastics would be closely related to the changes in the soil induced by microplastics. Our results suggested that microplastics could affect plant performance directly by changing the soil environmental factors, or indirectly by influencing the microbial communities. This study revealed the potential molecular mechanisms of maize's response to 5 types of microplastics. The results showed that although the growth characteristics of maize showed limited changes under certain microplastic treatments, maize leaves showed oxidative damage and molecular disorders. Multiomics analysis showed that under microplastic stress, maize's secondary metabolite biosynthesis, photosynthesis, energy metabolism, and carbohydrate metabolism were regulated. In addition, changes in underground characteristics caused by microplastics may have an impact on plant traits and functions.